How does the reference electrode work in EEG diagnostics? Imagine trying to listen to a conversation in a noisy room. To hear clearly, you need a baseline to compare sounds against. That's exactly what a reference electrode does in electroencephalography. It helps us understand brain activity by providing a stable point of comparison. In EEG, electrodes are placed on the scalp to pick up electrical signals generated by neurons firing in the brain. These signals are tiny voltage changes, often just a few microvolts, so they need to be amplified and carefully measured. The reference electrode acts as a kind of anchor point, giving us a 0 point to measure from. Without it, the signals would be confusing and hard to interpret. The reference electrode is usually placed on a part of the head that's less active, like the earlobe or the top of the head, at a spot called the vertex. This placement helps minimize the influence of brain activity from the area of interest. When recording, the EEG device measures the difference in voltage between the active electrode and the reference electrode. This difference is what shows up as the electrical activity we analyze. Think of it like measuring the height difference between 2 points on a landscape. The device uses a differential amplifier to subtract the reference signal from the active signal, giving a clear picture of brain activity at each site. There are different ways to choose a reference in EEG. One common method is the common reference, where a single electrode, often on the earlobe or the scalp center, is used for all channels. Another approach is the average reference, which calculates the mean of all electrode signals and uses that as the baseline. This method assumes that the total electrical activity across the scalp sums to 0, giving a balanced reference point. Some advanced techniques use virtual references, which are computed after recording, allowing for more flexible analysis. Choosing the right reference electrode is important in brain mapping and diagnostics because it influences how the signals are interpreted. For example, if the reference is placed too close to an active area, it can distort the data. Conversely, placing it far away helps isolate the activity of the region being studied. This flexibility allows clinicians and researchers to focus on specific brain regions or overall brain activity patterns. In neurotechnology, the reference electrode is a key component that helps improve the accuracy of brain activity measurements. It allows for detailed mapping of brain functions and aids in diagnosing neurological conditions like epilepsy or sleep disorders. Advances like virtual referencing make EEG analysis more adaptable and precise, opening new doors for understanding how our brains work. So, in simple terms, the reference electrode is the baseline that makes sense of the brain's electrical signals, helping us see what's really happening inside the mind.